This is a production of Cornell University.
So thank you. Today I'm going to be talking about the chemical language of fungi. So indeed, as Gary was basically telling you, we spent a lot of time, my whole life, honestly, that is my career, on working with fungi and specializing actually quite a bit on the signals that fungi make, a lot of them being what are known as secondary metabolites. These are compounds that most people actually know about, even though they don't know they know about it. Many of the pigments that fungi make, and you can see in the spores in this particular slide here, are melanins that often are natural products of a certain chemical class. Oh, I see it. Go over to this slide. So when we think about communication, and really this entire lecture is now about communication, fungal chemicals, this is something we learn when we're, I don't know, in first grade or preschool. We have eyes, ears, nose, mouth, and touch. Well, what do fungi have? Fungi actually share a lot of perception as us. They don't have a nose per se or a mouth per se, but they do perceive chemicals much the same way that, analogously to, let's say, people with their smell and taste. Fungi do have vision as well, and they do have touch responses. I'm not sure about hearing, but today we're going to talk about chemicals. So most of the chemicals that fungi make are known as natural products or secondary metabolites. And these compounds are all derived from primary metabolites. For example, we can have acyl-CoAs that all organisms make. They go to make fatty acids, for example, or membrane sterols. Fungi and bacteria and plants, I could say as well, can take these natural, these acyl-CoAs, they use them for primary metabolism, but they also incorporate them into two big chemical classes called polyketides, and through the mevalonate pathway into terpenes. And then amino acids, which of course fungi are going to use just as we do for our canonical proteins. They also will take these amino acids and they will make short peptides. They are known mostly as non-ribosomal peptides, although in our lab, we found in a collaboration with Frank Schrader's lab at Cornell, we characterize a new chemical class in fungi called isocyanides. Now, these are the typical secondary metabolites people now know pretty well. Honestly, this is pretty well known by many people. There's big efforts for mining fungal genomes to get these for new drugs. And in fact, we do quite a bit of that in our lab as well, though I won't touch on that aspect of our research. All of these complex natural products here are almost invariably found in a biosynthetic gene cluster. And this arrangement has been very useful in the bioinformatics study of fungal natural products, because typically in biosynthetic gene clusters, you'll have the synthase that makes the particular chemical class. For example, polyketides are produced by polyketide synthases. Terpene cyclases or terpene synthetases make these compounds, and non-ribosomal peptide synthases are responsible for making these metabolites. The amino acid sequence in these synthases is pretty conserved, so you can actually find them quite easily using various algorithms. Now, in a biosynthetic gene cluster, there's also typically 2 to 15 tailoring enzymes. And these will be enzymes, they're very critical too, because they'll take that backbone that's derived from, let's say, the polyketide synthase and be responsible for adding different moieties, such as, let's say, a methyl group, or we'll put in a polymerase and change or an oxygen group. So these are really important And a biosynthetic gene cluster will often, but not always, have a transcription factor that's specific to activate the expression of the other genes in the cluster. There will often be a protective gene because some of these compounds are antifungal. A good example is gliotoxin made by Aspergillus fumigatus. It is an antifungal. So the fungus itself has to find a way to protect itself, and it has an enzyme in the pathway that actually will cleave the gliotoxin to a more harmless compound. And there'll be hypothetical genes. So these are our canonical secondary metabolites that are produced from biosynthetic gene clusters. But not all of our secondary metabolites are coming from biosynthetic gene clusters. And today I'm going to present two of our latest stories, all involving secondary metabolites, but one of them is more simple. Our first story is a story of self-communication, somewhat analogous to the paracrine or autocrine signaling that some of you might be familiar with, let's say, in our in immunology. So fungi have to know when to develop. You know, you'll have an Aspergillus species, you have a small resting spore, it swells, it has to germinate. The germline then forms, then it has to branch, and you get heavy branching, and then you'll get development of the kinidiophore. So I'm going to be talking about a small metabolite, a secondary metabolite called an oxylipin that actually is a signal for branching. The second story is going to turn to the more canonical natural products, and not just fungal biosynthetic gene clusters. Oops, sorry, this is a very finicky little mouse here. But also will involve some bacterial natural products and the crosstalk between fungi and bacteria. But let's start with this first story on branching in fungi. This started long ago. This it has its roots in plant pathology, as does story 2, which I think is fitting for the audience. Many years ago, I got really interested in understanding how the seed pathogen Aspergillus flavus invades seed. Aspergillus flavus produces aflatoxin, which is the most potent carcinogen known to humankind. And you might think of all aspergilli as being, oh, they're saprophytic, they'll grow on anything, but actually that's not really true. So aspergillus are not too, they preferably like certain, I would say, crops. Flavus grows on seed, and it prefers oil seed. And I became really interested in trying to ask, why is the oil seed the preferable crop? At the time, I was familiar with some plant studies where certain fatty acids, oxygenated fatty acids called oxylipins, were involved in plant defense response to various pathogens. And these oxylipins are considered secondary metabolites, although they're simply made by enzymatic action on unsaturated fatty acids. Well, at the time I started this study, I became aware from another lab that Aspergillus species make these oxylipins, which are sort of similar, somewhat in structure to the plant oxylipins. So and they were already named by the lab that found them as psi factors. That name isn't really important. They use that name because it was precocious sexual induction. So it was known that Aspergilli made these, and they were involved in development of the fungus. I was interested in finding the enzymes that made the psi factors. And, oh, wait, let me ask, can you see the arrow when I move the arrow? Anybody?
Yes.
Okay, great. So I knew that the plant enzyme that made these oxylipins for plants were lipoxygenases. So I found in the genome of the fungus several, my student actually, Dimitris Tsitsigiannis, is the one who really was great in finding all of these oxygenases called PPOs, and they would make, and we found out that they made these oxylipins and aspergillus. Now, I'm not going to go through all this. We had many, many papers, but the bottom line is this, is that we found that these PPO enzymes that make the oxygenases, if you would delete them or overexpress them, you would actually change the developmental program of the fungus. Here's wild type Aspergillus nigelans. If you deleted PPOA, you got much more asexual growth and no sexual. Whereas if you deleted PPOC, you would get the sexual development. This is a Cleistothecium, the sexual fruiting body of Aspergillus nigelans, and less asexual spores. We also went on to find that when you added plant oxylipins to fungal cultures, it would change the development of the fungus. And when we would take human oxylipins, because humans make prostaglandins and leukotrienes, very similar to another type of oxylipin, they would also change the developmental plan of the fungus. So we ended up hypothesizing and putting forth several papers that we felt that oxylipins were signals, cross-kingdom signals, that acted in a quorum sensing manner with fungi to change development. Now, we could only get so far with all this research because something that held us back is that we were unable to procure enough of the fungal oxylipins. No company sells them, and it's very, very hard to purify enough of them to do very good work with fungi. However, there was a breakthrough that occurred. And now I'm going to take you right into the depths of story one. But I'm starting with a map of the world because story one has a COVID story that I thought, considering the times, you might be interested. This paper on where we actually have found how one of the fungal oxylipins actually promotes branching in fungi is a first author paper of my student. I'm thinking of COVID and what she went through. Min Yao, who graduated, okay? She is graduated and she did great. What happened is she submitted her paper to Nature last Christmas. And of course we knew if it even went through, we'd have to do gazillion more studies, tests to actually get it published. So she asked if she could go home to visit her parents for Chinese New Year. I said, sure, why don't you do that? So she goes in January, what's happening here, and couldn't come back because the COVID situation in Wuhan, though she did not live in Wuhan. She went in January and she was only able to get back to Wisconsin in July, 2 weeks before her PhD defense. Meanwhile, we got the paper back. Nature said, no, we like it, but why don't you, it's not quite Nature. We're going to pass you on to Nature Communications, which we said, okay, that's a good one too. And of course, we had so much work that had to be done in the laboratory, and there was Min Yao in China. So we actually, very proud of my lab. She communicated with everybody in the lab. And as a community, they got this paper together and it's been published in Nature Communication. Anyways, what a world we have. So the highlight is, and let me show you how we got to this, is that she found, and the many people on the paper, a specific oxylipin that Aspergillus makes, and its specific job is to induce branching. So how did she find this? One thing we did is we were very particularly interested in the PPO oxygenases because when you delete PPOA, you have a phenotype with the fungus. So she grew up the fungus and she asked, what oxylipins do we find? So PPOA makes two oxylipins, one called 8-HODE, this is derived from linoleic acid, and one is 5,8-dihode. And when she looked at fungal biomass and the supernatant, she could see that 5,8 was the most dominant and it was also secreted. So we thought, oh, well, let's see what 5,8 does. We were able to get MIGs of 5,8 because another lab had been able to purify it and express it in E. coli. So we were ready to go. And what does 5-8 do? You can see, I have some nice movies, but they were so big. I'm just going to show you these shots here. As you add more and more 5-8 into the culture of the fungus and at physiological levels, you can see there's an increase in branching. And here we have the branches for 100 microns of length of hyphae. We also found that you could visualize this, here you see here, by staining with chalcofluor white, which stains the chitin, and you could see there's more septed too. Commonly, when you have a branch, not always, but commonly you'll have a septa laid near. And here you can see that indeed we had a smaller septal distance when you're treated with this metabolite. We then asked, well, are there other compounds that could induce branching? There had been published in Nature about 15 years ago, the finding that a plant secondary metabolite called a strigolactone, and this is just a variation of the strigolactone, will induce branching in arbuscular mycorrhizae, but just in those fungi. And so we thought, well, we better check it because we hadn't found another chemical, and it did nothing. It actually did not induce branching in Aspergillus. That seems to be specific for the mycorrhizae. So what we next did is we bought or had our collaborator, Ernst Ohle, send us minute amounts of different oxylipins. And what you see here, these top oxylipins are all vertebrate oxylipins. And then these three right here are from fungi. This one is from Magnaportha, Aspergillus, and another Aspergillus one. And what we found of these oxylipins, we looked at the branching. Okay, didn't want to get to that right yet. The branching was specific to these fungal oxylipins, and more than one oxylipin could induce branching. It perhaps, we still don't know exactly if it's due to the 2-hydroxyls, but this is really interesting. So specifically because we knew other fungi made these, and Magnaportha grissii, the leaf rice blast fungus, made 5-8, we asked, well, is this branching just a response that only Aspergillus fumigatus makes, or are there other fungi? So we looked at a few other fungi, and we found that one other pathogen, Aspergillus flavus, also significantly branches when exposed to the 5-8, but the other fungi we looked at, and we didn't look at that many, did not. Including Magnaportha, we didn't see a significant difference in branching. However, we were bowled over because anytime you added 5-8 dihode to Magnaportha, you immediately got Aprosoria formation. And for those of you who are plant pathologists, you know right away that Aprosoria are the structures that Magnaportha needs to penetrate plants. Something we will be looking at further. So we found that this compound actually was a developmental signal for many fungi. Because we knew that oxylipins were perceived by G protein coupled receptors, the next thing we did was take a library that we'd made some years ago in Aspergillus flavus, where we had deleted the canonical G protein coupled receptors of Aspergillus flavus. So if you look right here, you can see again with wild type, when you added 5-8 dihode to Aspergillus flavus, you had an increase in branching. And we tested all of these G protein coupled receptors, and we found 3 where there was not a response or not a significant response to the 5-8. What was particularly interesting that 2 of these G protein coupled receptors, GPRC and GPRG, we had shown earlier, many years earlier, to be necessary for responding to plant oxylipins. So we believe that these particular receptors on the surface of the fungus are involved in perceiving the fungus's own metabolite. now we were fortunate enough, as I say, to have enough of this 5-8 that we actually went to where we really wanted to go in the first place, and that was RNA-seq. Actually, I had no idea what RNA-seq was when I was starting the study, because it wasn't, had not been, when I started working with oxylipins, because it had not been a technology at the time, but it's a very powerful one, as you know. We found that treatment with 5-8, and we did a 30-minute and 120-minute study, because we thought it's a signal, things are going to happen right away, and they did. We had many, many genes that were significantly differentially regulated, enough that we decided what we would do is just look at the transcription factors that are differentially regulated. This is all in Aspergillus fumigatus now. We found 35 transcription factors that were differentially regulated, significantly differentially regulated. Now, 35 is a lot, but luckily there is a transcription factor deletion library in Aspergillus fumigatus, headed by Dr. Michael Bromley. And so Michael joined our study, and we tested these 35 several times. And we came down where Minyao found that there were actually 3 of these transcription factors that, when deleted, did not respond to 5-8. And you can see one, for example, right here is one. This one did not respond. This one barely responded. These 2 did not respond. And in fact, they actually had an abnormal branching phenotype. So after finding these 3 genes, she actually went and extracted RNA again, just to confirm the RNA-seq results. And you can see that 2 of the genes, I just have the transcription factor names, HGO5O010, was induced by 5-8. This one was induced by 5-8, and 2 were repressed by 5-8. So what were the phenotypes when you looked at them? These were the 5 deletion strains that did not respond normally to 5-8. So here's wild type, the ethanol carrier, and the 5-8, and you can see the higher branching. And this particular mutant, you can say it was abnormal even as without treatment, and it did not respond. It was a hypobrancher, and hyperbranchers. So we became really interested in exploring these a little further, especially the 2 that had the most significant response. And what we did, so let me just go back here again, is oftentimes with a transcription factor, if you overexpress it, you can have the opposite phenotype as when you delete it. Not always, but sometimes. So we said, well, let's take this strain, which when you delete it has a hypobranching, and see if we get a hyperbrancher. Or likewise, what if we take a hyperbrancher, and when we overexpress it, do we get a hypobrancher? Because we reasoned that if you had the exact opposite phenotype, you actually would find a transcription factor that was very key in regulating branching. And we found one of those. What we found is this particular gene 082490. When you overexpressed it, let me just go back again. This is our favorite gene. When you delete it, it's a hypobrancher. And when you overexpress it, it's a hyperbrancher. So we are very thrilled to find this transcription factor through this methodology. It turns out it had been given a name before as ZFPA, although there had been no research really focused on this. It was just given that name from a microarray study from another lab. And so what we proposed here, we end the study, is we have found, to our knowledge, the very first transcription factor that regulates branching in fungi, and that it is a downstream target of fungal oxylipins, where the signal is mediated through a G protein, 1 or more G protein coupled receptors. Furthermore, we believe that this is very important as not only an autocrine signal for the fungus itself, but likely paracrine signaling between microbes or host tissue that it grows next to. What we're doing right now, one of our big stories here and interests is we know that the immune system, this is a macrophage and a neutrophil, that they also make oxylipins such as prostaglandins and leukotrienes that are really important in inflammation. And we suspect and are now interrogating the question, are the oxylipins from the Aspergillus fumigatus impacting immune cell response, and are oxylipins from the immune cell actually impacting the fungus by controlling branching once the fungus inside? We're also really interested, we're inviting a Magna Portha person to work with us on asking, could the plant, the rice oxylipins, actually be involved and recognized by Magna Portha as part of the signal to make a aprosorium? So that is our story number one. Story number two is now going to move into our interests in natural product signaling between fungi and bacteria and the consequences of that signaling. We're turning to canonical secondary metabolites now. We actually have 3 projects in the lab. One where we're looking at the soil microbiome, or I could even say plant microbiome with plant pathogens. One, the lung microbiome with human pathogens, and then in a collaboration with Ben Wolf at Tufts University, the interesting cheese microbiome with the fungi and bacteria that grow in cheese rinds. For the sake of time, I'm going to focus just on the first one today. So how did this get started? We're looking at a bacterium called Ralstonia solanacearum, a very famous wilt pathogen of many plants, and then the mycotoxin producing Fusarium and Aspergillus strains. Okay. So this story actually started a little accidentally, as many studies do. When I first moved to Wisconsin, before the building that I now am lodged in was built, I shared a lab in the plant pathology building with Dr. Caitlin Allen, who is very a world leader on the bacterium Ralstonia solanacearum. And of course, we were working with Aspergillus and Fusarium, and the story goes that sometimes there'd be a little contamination of an Aspergillus spore that went into Ralstonia plate, and things happen. You could see that the 2 microbes responded to each other. Many years later, we know what's happening between these organisms, and the response of these organisms to each other is in part mediated by 3 secondary metabolites. A lipopeptide we call ralsolomycin, made by the bacterium. A non-ribosomal peptide we call imisoquin, made by Aspergillus flavus. And a polyketide made by Fusarium species called bicaverin. I want to put a shout out. These papers are published, so I won't go into them into great detail. A shout out to our excellent collaboration with Dr. Frank Schrader's lab on these projects. So aside from these natural products, there is also a visual response when these organisms grow each other. And this is really important for this story. Here we have Ralstonia growing, and here we have Aspergillus flavus growing. And if you look at this particular plate where they're very close to each other, you can see that there's these bumps all over the mycelium of the fungus. These bumps were so big that you can see it with the dissecting scope, and maybe even without the dissecting scope if you can squint. What we found is that the fungus was producing chlamydospores when it grew near the bacterium. A chlamydospore, for those who are not familiar, is a very large resistant spore to environmental stresses. It can either grow within the mycelium intercalary or terminally. It's characterized by a very thick cell wall with a lot of chitin in it, and usually has a lot of nuclei in it. Well, we were really pretty surprised to see this because Aspergillus is not known for making chlamydospores. Some fungi make a lot, but most really weren't known. So when we found this, we thought there was something that the bacterium was producing that was involved. The other thing, and we'll get to that in one second, but the other thing we became really interested in was, wow, these chlamydospores are so big, could the bacteria possibly be going in? So we got a GFP labeled strain from Dr. Allen, and we found that indeed the bacteria would induce the chlamydospores and then go inside. And this is a movie of a Z stack. We saw some chlamydospore had hundreds, some had thousands of bacteria in it. So how did the bacterium induce these chlamydospores? That became our next query. And let's see here, let's go to the next slide. We found that it was due to a lipopeptide that the bacterium made. I'm not going to go through all the story on this. It's published, but the bacterium makes a lipopeptide called ralsolomycin. It has antifungal effects at high concentrations, and it induces the formation of the chlamydospores, and the bacterium then enter the chlamydospores. If you delete the genes in the bacterium, so you no longer have the lipopeptide, there's no antifungal effect, and the hyphae is nice and pristine with no chlamydospores. This seemed to us at the time that perhaps not necessarily something that was helpful for the fungus, or, oh, well, I guess you could, it was 6 of one, half a dozen of the other. There's that antifungal effect, but chlamydospores are spores that are very resistant to abiotic stresses, cold, heat, etc. But if a lot of bacteria go in it, is that going to obfuscate the, I would say, the advantage of making these? So we wanted to ask, what does the fungus do when confronted with ralsolomycin? And here again, we actually did another RNA-seq where we grew the fungus either with ralsolomycin present or without ralsolomycin, and we looked for genes that were significantly up or down regulated. And one was very interesting to us was a secondary metabolite gene cluster that is normally never expressed to high degrees in the fungus, and it was, to some degree, but it was down regulated by ralsolomycin. We were able to characterize this as this non-ribosomal peptide called imizoquin. And to make a long story short from this particular paper, we found that imizoquins are actually important for quick spore germination in Aspergillus, and they inhibit the growth of ralsolomycin, whereas ralsolomycin made by Ralstonia actually inhibits the expression of this compound that inhibits the growth of the bacterium. So this is a really interesting antagonism between two natural products that respond to each other. In this case, it seemed like perhaps in favor of the bacterium. Now, as we were working on this, we noticed that in the Fusarium species, the chlamydospores, which are smaller than Aspergillus, but many of them, all turned red. Just about. Here's one that's not red yet, but it will turn red. And here's the bacterium. It also enters inside this chlamydospore. The bacterium goes in all chlamydospores. So we wondered what this red compound was, and we were able to do some imaging and found out that this compound was a fungal polyketide known as bicaverin. It had already been discovered, but nobody had shown that it was responsive to ralsolomycin. You can see here that this treatment where we have the fungus and the bacterium, the bicaverin is really produced in great amounts right at the edge where the chlamydospores are formed. What we found is that opposite of Aspergillus that, see, sorry, this keeps flicking very quickly here. We found that this bicaverin gene cluster is actually induced by Rausolomycin. And we thought maybe this was giving the fungus some advantage. So we deleted the bicaverin genes in Fusarium. And then we looked at Ralstonia growth in confrontation with wild-type Fusarium or Fusarium that lacked bicaverin. And you can see that actually the bacterium grew better when there was no bicaverin. This one is a little complex here, right here. Oh my goodness gracious. We also showed that Rausolomycin induced, as I said, bicaverin production in Fusarium. One thing that's really interesting is bicaverin is always known to be inhibited by high nitrogen, but if you add Rausolomycin, it will allow the fungus to make more bicaverin. And here you can see the bicaverin genes are induced when you add Rausolomycin. So we went on to show then that bicaverin, oh, I showed you this a little earlier, that bicaverin reduced Ralstonia growth. This is with whole extracts of the fungus, but we also got purified bicaverin and showed that purified bicaverin actually reduced bacterial growth. So we concluded from this particular interaction that yes, in this case, Ralstonia induced the chlamydospores in Fusarium, just as it does in every fungus, and the bacteria would enter. But in the case of Fusarium, it actually would induce the production of a polyketide only in the chlamydospore where the bacteria entered and reduce the population, the number of bacteria. So these studies are all published. And I want to end, though, with a continuation of this work by another student in the lab, Nandita Venkatesh. So just to reiterate again, I think I'd mentioned that Ralstonia, of course, is a wilt pathogen, but so is Fusarium. So one thing that Nandita has been looking at is, well, what happens when you have these two pathogens together, knowing that Ralstonia can actually enter a chlamydospore of Fusarium, at least in the laboratory. And what is bicaverin doing? So this is work that is in preparation by Nandita. One thing that we found is that when you have Ralstonia, it does kill tomatoes immediately. Fusarium oxysporum is a slower killer, but it will eventually. But when you mix them together, it looked more like a Fusarium phenotype. And what she has found, and we're still doing some work on this, is that for reasons yet to be explained, when you mix Fusarium and Ralstonia together, you actually reduce the high severity of a bacterial wilt disease, but it is a little bit more than the fungus by itself. And then she went ahead to do a co-culture with Ralstonia and the bicaverin mutant Fusarium. And there's actually a bit more wilt. So it appears that bicaverin actually is reducing to some degree the severity of wilt disease by Ralstonia. We then, she's not quite finished with this yet, but we started to ask, well, okay, is there any benefit to the bacterium when it's in the soil, where presumably it is with the Fusarium? And so we have found that in this little mini winter study that the Ralstonia does grow better when Fusarium is there, which we hypothesize is because of the ability to enter and survive in the chlamydospores of the fungus. Now, to really get deep into understanding what's happening with this interaction with the bacterium and the chlamydospores, Nandita has gotten really much further, and we're very shortly to submit this work. Here is just a time lapse where you can see the chlamydospores forming in fungal mycelium when it's added to when raw solomycin is added. Nandita found it takes minimal amounts of raw solomycin, way less than the bacteria makes, to induce these chlamydospores. Here you can see it, way down at this molarity, we still get significant numbers of raw solomycin, of chlamydospores, and the bacteria makes a much higher amount than this. So we first wanted to see, okay, how many chlamydospores do you get and what molarity of raw solomycin do you need? And then what Nandita did is she developed a Z-stacking methodology so she could see the bacteria going inside the chlamydospore. And why she did this, you can see right here, is we had a hypothesis that, if raw solomycin is in the soil, could other bacteria get inside the fungus, or is it limited to Ralstonia? And she found a lot of bacteria could go inside the chlamydospores. Here we have Pseudomonas aeruginosa, opportunistic human pathogen, and Herbaspirillum, which is a nitrogen-fixing bacterium. What she did is went through and examined as many gram-negative and gram-positive bacteria that we could find from researchers in the lab. And we have a little story here. She found we were able to look at 3 gram-positive. They never enter the chlamydospore. And then she looked at 5 different gram-negatives, and all except for this one bacillus will enter the chlamydospore. So it seemed to be that a broad range of these bacteria. And what was interesting to us is when she looked at these and looked at a phylogenetic tree, the fungi that went inside the chlamydospore were never known to be endofungal bacteria before. So in red are the known endofungal bacteria, but we found that Ralstonia, Herbaspirillum, E. coli, and Pseudomonas also went inside. We believe this is the first time there's evidence for these fungi to enter bacteria. So is this something that's just limited to raw solomycin? Nandita looked in the literature and she found that there's many lipopeptides, and here's just some of them, that have been reported in the literature to induce chlamydospores. So we started to think, could this actually be widespread? And what we were able to do, she was able to do, is get a isolate of bacillus, the wild type that made fungicin that had been shown to induce chlamydospores, and one that was deleted for producing fungicin. And we found that indeed, when you had fungicin, and this happens to be with A. flavus and Pseudomonas aeruginosa, we get chlamydospores, and Pseudomonas enters inside the chlamydospores. This is getting very exciting to us. There's a lot going on here. Maybe this is very common. There's a lot of lipopeptides, a lot of bacteria, a lot of bacteria going inside. The next thing she wanted to ask was, well, will a chlamydospore only house one bacterium, or could there be more than one bacterium? So this is a case where she either co-cultured Ralstonia and Aspergillus alone, or Pseudomonas and Aspergillus, or both Ralstonia and Pseudomonas. And what we find is they both go inside. She did note that when they were introduced, sort of would change the ratio of how much each bacterium. So here we have a situation in E where we just had the fungus and Ralstonia, and she waited a certain time period and found about 15% of the chlamydospores were colonized. When she co-incubated Ralstonia and Pseudomonas, she found, and now she's just counting Ralstonia, that there was an increase in the number of Ralstonia that were inside the chlamydospores. She then did the same experiment, but she allowed Pseudomonas to arrive at the fungal court earlier than Ralstonia. And when she did that, we saw an increase in the number of Pseudomonas. So we think that obviously more than one bacterium can go inside the chlamydospore, but the relative amount of which bacterium is there might depend on which bacterium arrived first. The last thing she looked at is asked, does this colonization really, does it give any advantage to the bacterium? We had a little hint earlier that it did, but she wanted to look at this a little bit further under controlled experimentation. And here we have an example of the impact growing the bacterium with the funguses under starvation conditions. So what she did here, let's just look at the Ralstonian flavus interaction. She would grow the bacterium, either a wild type bacterium that made ralsolomycin or this one that does not make ralsolomycin, so no chlamydospores. And when she grew them in a situation where there was lots of nutrients for the bacterium, both the wild type bacterium and the bacterium lacking ralsolomycin grew equally with Aspergillus flavus. However, when she grew them under a situation where there was no nutrients, under starvation, you can see here that the bacterium that actually made ralsolomycin and could therefore go into chlamydospores grew much better than the poor bacterium without a lipopeptide. She found exactly the same thing with Pseudomonas aeruginosa. So this suggested to us that the bacterium actually does receive some benefit by being able to enter inside the chlamydospore. So I gave you a lot of what she has worked on. There's some other tests she did, but I would like to conclude with this second story right now by saying what we think is happening. We believe that this is widespread in nature, that actually chlamydospore formation in fungi is induced by lipopeptides that are made by many bacteria. It allows bacteria, one or more than one, to enter inside a fungal chlamydospore. It will protect them in different environments. And we think of the soil. We started with soil, but of course, all of the microbes we were working with are actually those that could be found in the human microbiome or in even other places such as the cheese microbiome. And we are now about, as I say, to submit this, that we believe that we should consider the chlamydospore as having its own bacterial microbiome inside it. So I will stop with those two stories. I do have more, but I'm conscious of the time here. So I just want to end this talk, show you what it looks like when there is no COVID virus at Madison, which we hope will come back again. I show in red the two people who really actually did most of the work, Minyao for the oxylipin branching signaling and Nandita for this really exciting bacterial fungal microbiome. Listed in yellow are those that helped Minyao during our COVID crisis. I also want to shout out to Frank Schrader, because a lot of the work that I mentioned, some of our earlier work, he's been crucial for helping. And with Minyao, also Dr. Oli Nupsala Sweden, who actually made some oxylipins for us, and Dave Beebe, who helped us with some microfluidic devices. And with that, I'm happy to take any questions.
Nancy, thank you very much. What a terrific seminar there. We really appreciate it. So I'm sure I don't see anything in the chat room yet, but I'd encourage anybody to put your questions in the chat room.
Hi, Nancy. Always proud of you. Today is no exception. Regarding the first story and the transcription factor induced by the oxylipin,
Yes.
do you have any idea about the conservation of that transcription factor across fungi?
We do have a little bit of an idea about that. It's honestly, we looked in Aspergillus flavus and it's there. So one of the next studies will be to see if it also regulates branching there. It does seem to be pretty good homologs in Ascomycetes. So
Okay.
it'd be really interesting to look at some other fungi to see what it does with them.
Yeah. And are there any other morphological developmental effects other than branching and possibly acrosorium?
Yes, there are, and I'm glad you asked that. What I didn't tell you is that this is a signal for inducing secondary metabolism too. We're really excited about that. We hypothesize, well, so a lot of the canonical secondary metabolites, we believe there's lots of evidence to suggest, well, some evidence that they're associated with the endocytic pathway. And we think they're associated with the growing tips and septa. And so we will be exploring that further. Calcium signaling too. There's a lot of calcium signaling genes that are upregulated.
Gary, are you seeing questions in the?
Yes, there are a couple of questions here. So you've got a couple of applauses, I should say. There's a question here, how, from Saeed, how will Ralstonia be freed from the chlamydospore? Can it easily go in and out, or does it need to await germination of the chlamydospore to escape?
Oh, that's an excellent question. And I don't have an 100% answer for this. Now, Nandita has sometimes, but sometimes not, seen as the fungus germinates the bacterium traveling through the hyphae. Not all the time, but sometimes. I suspect that this does happen on occasion. So when that chlamydospore germinates and, oh man, you like get 10 to 15 germ tubes coming out of it. It's so big that the bacteria can go out that way. Maybe it can also go out through cell wall remodeling too. With that, we don't know yet.
So a follow-up question there from Raleigh is any ideas on how the bacteria enter the spore, and then can you speculate why gram-positives cannot gain access?
Yeah. So we have some ideas that we want to follow up on. I don't know if Nandita will, she does want to graduate, but we're wondering if some of the type 3 or some of the secretion systems in the bacteria are important. So gram-positive and gram-negative have different secretion systems. Maybe that's part of it. The gram-positives, I don't think this is why it stops them from going inside, but they do. We have some nice pictures where they like to stick on the hyphae on the outside of the fungus, but that shouldn't preclude them. We're thinking it might have something to do with these secretion systems of the bacteria.
Great, and Maria Harrison asked a very similar question. Stephen Higgins said you get a great talk. Obviously not all Ralstonia enter the chlamydospore. Are there any nutrient signals in these spores that attract some Ralstonia over others?
We don't know that. One thing we want to ask in the future is the chlamydospores have a lot of lipid bodies in them. They're just chock full of lipids. And we're guessing the bacteria might be living off of the lipid bodies. So we want to, there's several ways we could address that question, but we don't have anything specific right now.
I don't see any other questions in there right now. I want to remind you, Nancy, that your talk has been recorded. So we'll be sure to get you that recording.
Okay, great.
Used for other occasions.
Okay, well that sounds wonderful. And I'm sure I know I'm meeting with some of the postdocs and grad students.
Yeah, so I see that you're right on the button there. It's supposed to start at 1:45, but why don't I suggest that you'll need a little bit of a break and then ask the students and postdocs to rejoin you at the Zoom link that was provided to them.
Okay, so, sorry, I think I went over there. I will join postdocs and grad students very shortly.
So let's give a round of virtual applause for Nancy here. Thank you very much, Nancy.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.